There we go.
Yeah, recording.
And thanks also to Sepide for the invitation to share some of our work on the interleukin-11 signaling complex and structural biology of some of the inhibitors that we've been working with. Now I'm just going to tuck everyone away out of the way so that I can see all my slides. And just before I get into it, I want to acknowledge Riley Metcalf, who was an exceptional PhD student in my lab and really drove pretty much all of the work that I'm going to be talking about this morning. So Riley's moved on now. He's now in the US at the NIH, but of course continues to do wonderful things. And he's actually just been offered a new position at the NIH, which I think is really exciting for him. Excuse me. So Riley was also ably assisted on the cryo-EM side by Eric, who's obviously a familiar face to a lot of people here. So a huge thanks goes to Eric for facilitating that side of the work. And finally, just a declaration from me that I'm a scientific co-founder of Nelcanon Therapeutics. And this is something that's been a really exciting development over the last couple of years, even if it's also been thoroughly daunting. So cytokines are small signaling proteins, generally less than 20 or 25 kilodaltons or so. And these send messages between cells and sometimes within the same cell. So cytokines generally signal through multimeric signaling complexes comprising receptor subunits that have fairly large extracellular domains and just a single simple transmembrane pass and fairly dynamic intracellular regions. And the primary downstream signaling from cytokines is via the JAK-STAT pathway, although other pathways can be activated, such as the ERK pathway. So signal-transducing receptors have Janus kinases, or JAKs, bound to their intracellular domains, and these become activated by phosphorylation when the cytokine binds. So the activated JAKs then phosphorylate STAT molecules. These are transcription factors, and these can be translocated to the nucleus, where they regulate transcription of cytokine responsive genes. And amongst these genes are the SOX proteins, the suppressors of cytokine signaling. And these regulate further activation of the pathway, but also degradation of the signaling complex itself. So there are two main mechanisms by which formation of the signaling complex is proposed to result in the activation of the bound JAKs. The first of these is a simple cytokine-mediated receptor dimerization model, where the cytokine binds to a single receptor subunit. This binary complex then recruits a second receptor, and this brings the JAK molecules close enough in space to phosphorylate each other by transphosphorylation or perhaps by autophosphorylation, thereby mediating the downstream signaling. The second mechanism involves a preformed receptor dimer, and the cytokine actually induces a conformational change in this dimer, which changes the orientation or positioning of those JAK molecules, leading to their trans or autophosphorylation and therefore downstream signaling. And this is a bit of an outstanding question in the field, which of these mechanisms, or indeed which combination of these mechanisms, is relevant to which receptors. And this is something that we're really interested in and we are working on, but I won't be talking at length about this work today, just touching on some of the initial dynamics that we see in our receptor complex. And the family of cytokine receptors that we're most interested in is the interleukin-6 family or the IL-6 family. And this is one of the largest cytokine families with about 15 unique cytokine signaling complexes, depending on how you count. And I'm just showing a few representatives of the family here. So the unifying feature of these cytokines is that they use GP130 as a signal-transducing receptor subunit. And so this family actually overlaps with the IL-12 family of cytokines, some of which can also use GP130 as their signal-transducing receptor. But within the IL-6 family, there are two major classes. There are cytokine receptor complexes that contain two molecules of GP130. So the active signaling complex comprises two molecules of GP130. IL-6 complex is an example of this, and the IL-11 complex is also an example of this. So the second class signal through complexes that contain just one molecule of GP130 and one copy of another cytokine-specific signal-transducing receptor that also has a bound JAK molecule. So the leukemia inhibitory factor receptor is an example of this, as is the oncostatin M receptor. So you can imagine that within this family, there's quite an array of interactions that GP130 can make with the various cytokines and the various receptors in these complexes. And we're really interested in the fundamental mechanisms of how these complexes assemble, but also in how we can interfere with this process to develop inhibitors. And the family member that we've made most progress on in this respect is interleukin-11 or IL-11. So IL-11 was originally recognized for its central roles in platelet production, but more recently, important pathological roles have been identified, including in autoimmune diseases such as arthritis, asthma, and inflammatory bowel disease. It drives fibrotic complications of the gastrointestinal tract, the heart, kidney, liver, and lung. And it also promotes the growth of a number of solid tumors, including breast, lung, endometrial, and gastrointestinal cancers. So IL-11 is really gaining quite a lot of interest recently as a therapeutic target. So IL-11 signals by binding to its two cell surface receptors, the interleukin-11 alpha receptor or IL-11R alpha, and of course GP130. So this alpha receptor is not a signal-transducing receptor in that it doesn't have JAK bound to its intracellular domain, but it functions to define the cellular specificity of IL-11. So only cells that express the alpha receptor will respond to IL-11, whereas GP130 is much more ubiquitously expressed. So cytokines generally bind to their receptors at the margin between two fibronectin type 3 domains, and we call this the cytokine binding module. And GP130 is actually a member of what's known as the tall cytokine receptor group, which basically means that all of the cytokine binding action occurs in this top part, so quite a distance away from the membrane. And despite its importance, actually historically very little work has been done on these structural biology of these membrane proximal domains in the context of signaling complexes. But recently this has started to be rectified. So these two structures that we've solved of the cytokine and the alpha receptor in isolation, but of course we were most interested in how these come together to form the signaling complex. So we actually went a little bit overboard and solved three separate structures of the IL-11 receptor complex. We have the complete extracellular domains of the complex at a little under 4 angstroms resolution. This was solved by cryo-EM. We also have the cytokine binding domains of the complex. So just these top domains here, also solved by cryo-EM at about 3.5 angstroms resolution. And because I'm a crystallographer, I had to complete the set. So we also have a crystal structure of the cytokine binding domains. I jest, of course, because this was an example where we had been working on the crystal structure for quite some time and not making a lot of headway. Once we had an instrumentation, the cryo-EM structure seemed to drop out quite easily in comparison. And I should also point out that the X-ray data for this structure were quite anisotropic. So the information content of this crystal structure was actually lower than the cryo-EM structures, as much as it pains me to admit that. So this is a hexameric complex in all cases that contains 2 copies of each of the components. So we have 2 alpha receptors here in purple, 2 IL-11 molecules in green, and of course 2 GP130 molecules in this salmon pink color. And we spent some time confirming the stoichiometry and configuration of the complex. So we used sedimentation equilibrium analysis, multi-angle light scattering, and small angle X-ray scattering. And I show these data not because we didn't actually trust what we were seeing in the cryo-EM structures, but because we were able to apply these techniques to all the intermediate complex assemblies that I'll be talking about later on in the talk. So you can rest assured that each of the complexes that I will be talking about have been verified by multiple techniques. And using the analytical ultracentrifuge, we were able to show that the hexamer actually assembles in 3 sequential and independent steps. And what I'm showing here are size distribution analyses from sedimentation velocity experiments, where we have increasing sedimentation coefficient here, indicating a larger species in solution, essentially. And what this shows is that the binding of IL-11 to the alpha receptor is a simple 1-to-1 binding in the initial step, forming this binary complex between the 2 components. If we add in GP130, we get formation of this much larger complex, and this is consistent with that hexameric complex that we observed in the cryo-EM. But using our cryo-EM structures, we were able to see that the first domain of GP130 is important for forming that hexamer. So by removing that first domain of GP130, we were able to show that this trimeric intermediate comprising just 1 copy of each of the components is a stable and fairly high affinity intermediate in the assembly process. And we can use our structures to get some detail on the interfaces that contribute to formation of the hexamer, and there are actually 10 of them in all. So we have a unique interface that forms at that first step between the cytokine and the alpha receptor, and we call this site 1, being the first step. The second interaction to form that first trimer with the first molecule of GP130 is a composite interface that's formed both by the cytokine and the alpha receptor. And this binds to the cytokine binding module of GP130 in that second step. And we call these interfaces site 2A and site 2B. And the third interaction is also a composite interface, but now with that second trimer of the hexamer, where the D1 of that first molecule of GP130 interacts with the alpha receptor and cytokine of that second trimer. And we call these sites site 3A and site 3B. And of course, because the hexamer is symmetrical, these interfaces are all replicated once. So we have 10 interfaces in all. And we can explore these interfaces in more detail. So if we first zoom in on site 1 between the IL-11 and the alpha receptor, we can see that this is mediated primarily by this helix here and this long loop structure here. And we call this the AB loop. And this is a really important feature that I'll be coming back to a number of times during the talk. This is again important interactions between the AB loop here and the alpha receptor. Now moving to site 2A between the cytokine and that first molecule of GP130. And this is actually mediated primarily by a group of arginine residues that form electrostatic and cation pi interactions with the GP130 molecule. Some more detail here. And now moving to site 2B, between the alpha receptor and that first molecule of GP130, you can see again quite a highly charged interface between those two molecules at that site. Now moving to site 3A, and this site is actually dominated by this tryptophan residue, residue 147. And again, this is another feature that I'll be talking about at length later on in the talk. And now to site 3B, and this is a much smaller interface, but important nonetheless. So one thing that we also noticed from our structures was that the cytokine actually undergoes significant structural rearrangement upon complex formation. And this is actually quite unusual for cytokines. Normally cytokines, and in fact cytokine receptors, also show pretty limited conformational change, say for side chain rearrangements on complex formation. And what I'm showing here are the crystal structure of the unbound cytokine in pink and the structure and complex with the receptors in green. And what you can see is that this loop, this AB loop, that I've already pointed out, changes configuration quite considerably. And this is accompanied by a movement of this arginine side chain here. And we can see some more detail of this conformational change in detail in the structure here. So zooming in again on site 1 between the alpha receptor and the cytokine, what I'm showing here is in pink the crystal structure of the unbound cytokine. And you can see that the electron density actually supported the position of the loop pretty well. And there are clear reasons why that loop is in the position it is because of some surrounding interactions. What we can do now is morph between that crystal structure and the bound structure of the cytokine. And you can see this arginine residue reaches out and now forms hydrogen bonds with the backbone of that AB loop. And we think this is really important for stabilizing that AB loop in the new position that it finds in the complex. And out of frame here, what you can't see is that there's actually an unwinding of this B helix here to allow this rearrangement of this loop. So one of the things that we also noticed in our structure of the complete extracellular domains of the signaling complex was that the density became poorer further away from the cytokine binding regions of the complex that we got, so towards these membrane-proximal domains. And they definitely showed much more smeared density, in fact, to the point where it was actually very difficult to build into the density there. And we wondered whether this may be due to flexibility of these domains. So we used 3D variability analysis to analyze this conformational flexibility within our population of particles from the cryo-EM. And this analysis gives us a number of components to the movements. And I'm only showing 2 of these here, but you can see that graded from green to blue, that the legs, what we call the legs of the complex, actually move quite considerably, particularly in component A, where you have the swinging of them. In component B, we have a movement of those legs towards each other in this orientation there. And we can see this a little bit more dynamically in a movie. So in this orientation, we have very little movement. This is component A. However, if we look down the axis of the complex, you can see those legs move towards and away from each other. And in the cytokine binding domains, very little movement again. So this flexibility of the membrane-proximal domains of GP130 is potentially very important. Is there some really... I think that's fixed. So yeah, this flexibility is potentially quite important since we know that the relative geometry of these domains is critical for signal transduction. And this is presumably by coordinating the relative positions or orientations of those JAK molecules that are bound to the intracellular domains of the GP130. And we have done some work on hereditary mutations that occur at the domain interfaces of those membrane-proximal domains of GP130. And these result in complete loss of signaling, and they also appear to alter the dynamics and pose of the legs of GP130. And we're currently really interested in working on understanding the implications and consequences of this flexibility, both in the native form and in some of these mutations that are being increasingly identified in patients. And so the therapeutic potential of targeting IL-11 signaling has sparked a lot of interest in signaling inhibitors. And there have been various strategies that have been applied to generating inhibitors that are specific to IL-11 signaling. And these include generating neutralizing antibodies against the cytokine itself, but also the alpha receptor. But there have also been two IL-11 variants that have been reported in the literature to inhibit signaling, both in vitro and in vivo. And these have been around for quite some time, you can see, but really the understanding of how these work and in fact their functional efficacy has actually been pretty limited. So we were interested in how these might be working. So the first of these is the W147A mutation, which removes that critical tryptophan residue at site 3 that I pointed out a little bit earlier. So just to orient you here with this cytokine structure in this orientation, we have site 3 at the top and site 1 on this side. So by removing this critical tryptophan residue, we think that this mutant is interfering with hexamer assembly. That'd be a logical outcome of that. The second variant actually combines that 147A mutation with 5 consecutive mutations in that critical AB loop that forms those key interactions at site 1 with the alpha receptor that I pointed out. And this is where the native sequence AMSAG is mutated to PAIDY. And this variant's been dubbed IL-11 Mutein, presumably because of the number of mutations that it carries. But as I said, neither of these variants have been very well characterized in terms of their mechanism and function. And in particular, the role of these site 1 mutations is really unknown. So they were identified by phage display of random mutations in this area of the protein, but there was really no follow-up about their functional effects. So the first thing we wanted to do was establish whether these variants were in fact signaling incompetent, because clearly in order to inhibit, these variants must have no signaling activity of their own. And here what I'm showing is a measurement of phosphostat-3 as a readout of IL-11 signaling. And this is made using a phosphoflow assay after stimulation of cells with increasing concentrations of the cytokine variants. And we're using BAF cells here that express both the alpha receptor and GP130. And this is work that was done by Kai Feng over at Weihai. And you can see that the wild type IL-11 produces a very robust induction of phosphostat-3 with a really respectable EC50 in the cell system. And what we also found was that the W147A mutant retained quite potent signaling activity, albeit with a substantially higher EC50 than the wild type cytokine. And this suggested to us that actually it was probably not going to be very good as a signaling inhibitor. IL-11 mutein, on the other hand, showed no signaling, even at these very high concentrations, which we thought was probably a bit more promising. So the question then becomes, can IL-11 mutein act as a competitive inhibitor of IL-11 signaling? And it turns out that it can with a mid to high nanomolar IC50. So here we stimulated cells with IL-11, so wild type IL-11, and applied the inhibitor variants while still in the presence of the wild type cytokine. And so what this shows is that IL-11 mutein is acting competitively to bring signaling down to baseline. As expected, we saw little to no inhibition with the W147A mutation. But of course, we now wanted to know how mutein, the IL-11 mutein works in a mechanistic and structural sense. So as a starting point, we solved the crystal structure, and this is it here in blue alongside the crystal structure of the wild type IL-11 in green. And at first glance, the structure looks very similar to the wild type. But if you make an overlay, we can actually see some really important and interesting changes in the conformation of that AB loop. So we can see that this top part of the AB loop near site 3 has changed. And also there are changes in this bottom part. And if we look closely at the structure, we can see why this is. So there are particular loop core interactions that are mediated by residues in the AB loop with the alpha helical core of the structure in the wild type protein. And in the center of this, there's actually a hydrogen bond between this histidine 86, which is part of the helical core structure, and serine 53 on the loop. In the mutein, the same hydrogen bond is actually mediated by threonine 56. So essentially, the register of these loop core interactions that we see in the two structures is shifted by 3 residues. And the result of that is that the bulk of the loop is shifted upwards in this direction here. So we have more loop above the hydrogen bond here and less loop below the hydrogen bond here in the IL-11 mutein. And this is actually caused by those PAIDY mutations in the AB loop. And we think that the proline residue is key here because it constrains the backbone of the AB loop in that position in a limited configuration. Of course, being cautious crystallographers, we wanted to make sure that what we were observing was real and not some weird artifact of the crystal lattice or the crystal packing. So we did some molecular dynamics simulations, and these were done with the help of Craig Morton. And here I'm showing simulations for the wild type IL-11 alongside the mutein. And as you can see, the configuration of the loop in each case is pretty stable throughout the microsecond of simulation. And certainly the loop core interactions are stable. But what we did see was that that hydrogen bond between that threonine residue and histidine residue in IL-11 mutein was actually formed for a much larger proportion of the trajectory than the hydrogen bond in the wild type IL-11. And what this suggested to us was that actually this altered conformation of the loop in the IL-11 mutein may be more stable than in the wild type protein. And in fact, we were able to show that the thermal stability of the mutein was increased relative to the wild type protein. But we had to think a bit about what this altered loop conformation might be doing in the context of the hexameric complex. So clearly an inhibitory cytokine variant must alter formation of the hexameric signaling complex sufficiently to preclude signal transduction. And the hypothesis we came up with here was that the IL-11 mutein loop was helping to disrupt the interactions with GP130 at site 3. So our complex structure shows us that there's quite a neat and intimate contact between part of that loop at site 3 and that GP130 molecule of the second trimer. If we overlay the mutein structure now, we can see that this part of the loop on the mutein structure in blue will clash with the GP130 molecule, potentially creating a steric bulk at site 3 that precludes binding of that second trimer and removes essentially formation of the signaling competent hexamer. So this would be a long-range effect of the mutations which are located down here. And using analytical ultracentrifugation again, this is exactly what we saw. So I've already shown you that the wild-type IL-11 mediates formation of this nice, neat hexameric complex. In the case of the IL-11 mutein, the complex is formed as much smaller, and this actually corresponds to the trimer of one molecule of each of the cytokine, GP130 and alpha receptor. And we don't see any hint of any larger complex that might correspond to the hexamer. So while the W147A mutation alone was not sufficient to block formation of the hexamer and therefore signaling, adding the PAIDY mutations alters the conformation of the AB loop just enough to completely abrogate the binding at site 3. So in effect, the loop conformation is reducing the already weak binding due to the W147A mutation. So this told us part of the story, of course, that IL-11 mutein is blocking formation of the hexamer. It's stalling the assembly of the complex at the trimeric stage, but it didn't tell us how mutein was effectively competing with IL-11 for receptor binding. So one obvious potential explanation is that IL-11 mutein binds much more strongly to the alpha receptor than the wild-type IL-11. But we have isothermal calorimetry data that already told us that there really wasn't much difference between the affinities. So here we turn to surface plasmon resonance to look at the kinetics of the binding of IL-11 mutein to the alpha receptor. So we're looking for on rates and off rates. And this is work that was done with the help of Larissa Doughty. And what we saw was perhaps a modest increase in the affinity of the IL-11 mutein for the alpha receptor over the wild-type protein, but really it's only a little more than twofold, so not really enough to account for significant competition. But more importantly, what we saw was a slower off rate for the IL-11 mutein. So effectively, the IL-11 mutein has a longer residence time on the alpha receptor. At each binding event, it spends longer bound to the alpha receptor. And we think this is aiding competition with the IL-11. But what's causing this slower off rate? Normally, when you tamper with interface residues, you expect to impair the binding properties and not improve them. And effectively, what we're showing here is that the PAIDY mutations have reduced dissociation of IL-11 mutein from the alpha receptor. And here we think there are some more clues that can be eked out of the structures. And this is because the IL-11 mutein in the unbound state actually shows conformational changes in the AB loop around site 1 that are similar to the complex bound form of the wild-type IL-11. And the more observant of you might have already noticed this from some of the images I've already presented. But what I'm showing here is the configuration of the AB loop in the unbound cytokine in pink, the complex bound cytokine in green, and in blue, the configuration of the AB loop in the unbound IL-11 mutein. And you can see in this lower part here, the configuration of the mutein loop actually resembles the bound form of the wild-type cytokine more than it does the unbound form. And we also have the signature unwinding of the bottom of that B helix here. And looking closely again, we can see other similarities in that this arginine residue that stabilizes the position of the AB loop in the complex is also stabilizing the position of the AB loop in the unbound IL-11 mutein. And we think this probably also contributes to the stability of the AB loop that we saw in the MD simulations and that increased thermal stability that we observed. And what we think is going on here is that this new conformation is actually priming the IL-11 mutein for binding to the alpha receptor and perhaps contributing to that altered dissociation kinetics that we observe. So essentially IL-11 mutein is already in a configuration to bind, so it can stay there for longer. So just to summarize this now, signaling by IL-11 proceeds by sequential binding of the cytokine to the alpha receptor to form this binary complex that can recruit a first molecule of GP130 to form a trimer. The trimer can then dimerize with a second trimer to form the signaling competent hexamer, leading to activation of the JAKs and downstream signaling. So IL-11 mutein can also bind the alpha receptor and that first molecule of GP130, but once this trimer is formed, it can no longer go on to dimerize with a second trimer to form the hexamer, and therefore there is no downstream signaling. And we think that mutein competes with IL-11 via a longer residence time on the IL-11R alpha. And this has two effects. Firstly, a direct competition. But secondly, remember that the interaction of the IL-11 alpha receptor binary complex with that first molecule of GP130 is actually mediated by this composite interface. And we think that if mutein spends a longer time on the alpha receptor, there's actually a longer period that GP130 can come in to bind. And in fact, we do see a higher affinity of the IL-11 mutein alpha receptor complex for that first molecule of GP130 by ITC. So essentially, the mutein is increasing the affinity for GP130, that first molecule of GP130 also. And the end effect of this is that the alpha receptor and the GP130 available on the cell surface are actually bound up and sequestered in this stable trimeric intermediate, leading to competitive inhibition of IL-11 signaling. So we have this really beautiful and intricate interplay between the local effects of the PAIDY mutations on the cytokine conformation and the alpha receptor binding at site 1, coupled with these long-range effects on the configuration of the loop that reduce binding of the second trimer at site 3. So these mutations actually alter binding characteristics at each binding interface of the complex, which I think is really quite amazing and quite an elegant mechanism of inhibition. And I don't think that either Riley or I, when we started this work, envisaged that we'd find such a complex and surprising set of structural mechanisms of inhibition. And with that, I'd just like to thank all the people involved in the work. I've mentioned important names on the way through. Also, just to mention Tracy Potosky and the Potosky Lab, who really did all the work that forms the foundation and basis of all the work that we've been able to do in the structural biology realm. And they carry the cell biology of this project as well. Also to the Australian Synchrotron, the SACS and MX Beamline staff, Janet and Bevan at CSIRO-C3, which of course is not in existence anymore, and our funders. And thanks for your attention.
Thank you, Michael. So please feel free to have your questions.
I definitely did fire through that a little bit faster than I expected to, so hopefully it was all clear enough.
That's right. I also delayed your start too, sorry.
Maybe that's why I felt like I had to go through quickly. My video should be coming up soon. Here we go.
Yeah.
Hi everyone. I might ask a question.
Hi, Sarah.
Hello, nice presentation. Thank you for that. I'm a bit. I have a question, maybe more a method question.
Yeah.
I'm a bit intrigued to see your experience with crystallography versus cryo-EM. Can you maybe you mentioned it briefly that you had some anisotropy with your crystallography data, and I don't have much experience with crystallography and crystallography data, but I was wondering if you can comment on where you see the advantages or disadvantages of using cryo-EM. I think like you showed very nicely the 3dVA that you got from the cryo-EM data as well, which was quite interesting to see where the dynamics are. And do you think that's the advantage of like using cryo-EM for your complexes?
Absolutely. So, I mean, that's something that you just really can't do in an experimental sense with a crystal structure. You can run simulations, and there are various things that you can do based on B factors and all those sorts of things with a crystal structure, but you wouldn't be getting beautiful movies like we were able to produce. So I guess there are a couple of questions to answer there. So the crystal structure is anisotropic in that the diffraction resolution in the 3 dimensions of the diffraction data is different. So while that nominal resolution was 3.8 angstroms, in the other 2 directions of the diffraction data, the resolution was much poorer, and that causes a directional smearing of the density. And it's a little bit like favored orientation in the cryo-EM. And in fact, I have to admit that we also observed favored orientations in our cryo-EM data, but we were able to, with Eric's help, we're able to get around that using various techniques. The other thing is that we would never have been able to crystallize that complete extracellular domain of the protein. Essentially, we had enough trouble with just the cytokine binding domains, which are the more stable domains. So it would have been. I mean, I wouldn't even. Probably wouldn't have even embarked on a trial to crystallize the complete extracellular domain. So those were data that we were able to get from the cryo-EM that I don't believe we would have thought from crystallography. Again, these things are a little bit painful for me to admit as a historical crystallographer.
No, thank you. I was wondering, are you still using crystallography for, like, let's say, some applications where you work with smaller proteins so that you get to the higher resolution that would inform drug discovery maybe a bit in more like detail? Yeah, absolutely. And I mean, you may have noticed that one structure that we've been trying to get, which has been missing, is the mutein complex with the 2 receptors. That's something that might be doable by crystallography. It's probably a little bit small. We did try in the microscope, but probably a little bit small and too highly dynamic. But the answer is yes. We continue to use crystallography for a lot of the smaller components of what we're doing, as well as small molecule binding to various things. So yeah. Thank you. Thanks for your presentation. Yeah, thanks. Thanks for the question. Hi, Josh.
Hey, maybe I'll just go ahead and ask. Thanks for the talk. Really lovely structures, and I know it's really hard to get sometimes a nice homogeneous orientation of these receptors. I'm sure you'll have seen the 2 leptin receptor structures that came out very recently. Yeah. Was just wondering what your thoughts on those were, and how maybe they change your understanding of your structures and the similarities and differences there. And maybe on a methods angle, I noticed they saw like compositional heterogeneity in their cryo-EM data. So they have kind of complexes of dimers and trimers and hexamers. Did you see that as well in your cryo-EM?
I missed part of the question, but the answer to this second half is that we did see a lot of broken complexes on the grid. So the complex didn't like whatever was happening to it on the grid before it was frozen. But we didn't see higher order oligomers that have been seen, I know, for a couple of other systems. And it's something that we have looked into. So looking for higher order oligomers, so whether the hexameric structure can go on to be any bigger. But we don't see it by any of the techniques that we use. And of course, our most sensitive technique in that respect is the AUC. And as you probably saw, we've put it through the ringer with the centrifuge.
Yeah, I guess, sorry, can you hear me better on this microphone?
Yeah.
Good.
Yep.
Yeah, the first part of the question, thanks for that, was about, I guess, the leptin receptor structure, how that is related to this receptor and what you've kind of maybe learnt from their structures, or does it change your interpretation of what you're seeing? Or have I got the connection wrong and they're kind of distantly related?
I have to admit I haven't looked in detail at the leptin receptor structures, but I mean, one of the is that the complete with the intracellular domain as well?
No, it was just the one I was looking at was mostly the extracellular domains. There was two, one by Garcia's lab and one by another group.
Oh, the intracellular domain with the Jack.
No, it was extracellular. Yeah.
Yeah. No, sorry.
Okay.
Yeah,
it was very recent. But yeah, it did remind me a lot of the structure that you're showing with the multiple proteins. And yeah, obviously, yeah, still quite challenging in terms of resolution interpretation. But
yeah, well, so I mean, one thing that we want to get eventually is the complete structure with the intracellular domains as well. And of course, that I know is very challenging, and I know there are other groups in the world who are trying to do it. And you know, it may be that we need to engineer things in such a way that that's possible. But that is something that we want to get information about how these things are oriented. So one thing about the GP130 system that is not a feature of a lot of other cytokine receptor families is that there's both GP130 dimers and homodimers and heterodimers within the family. Yep. So there's got to be some promiscuity with the way the GP130 comes together, but also the dynamics of the GP130 on the cell surface. So there's various historical literature that says that GP130 is a preformed dimer on the cell surface, but that really doesn't make a lot of sense when it also has to dimerize with these other receptors to form these other receptor complexes.
All right, thanks. It's very exciting work.
Cheers.
I have a quick question.
Yep.
I know we are approaching the time, but so if I followed correctly, when you showed the molecular dynamics of the loop and the interactions taking place, was it like 53 and 56? In wild type it was 53, I guess.
Yeah.
In the mutein
so
it was 56.
Yeah, correct.
So, but those residues are present in wild type as well, right? The mutation is somewhere else.
Yeah, the mutation is actually outside those positions. Yeah.
So what I was wondering is that the molecular dynamics shows that the hydrogen bond between the 53 and that the other residue is quite dynamic. It's breaking and then reforming and breaking and reforming, which means that loop may be moving somehow, right? And you showed that the hydrogen bond with 56 was much more stable. So I was curious, if you can run the molecular dynamic simulations for much longer, would that loop at some point, once it breaks, shifts, and if it makes that 56 bond, would it then stabilize that conformation instead?
Yeah. Well, I mean, we ran a microsecond, which sort of felt enough for us, but the answer is yes. I mean, anything could happen. There are actually a lot of other interactions so around that hydrogen bond. So it's not just that hydrogen bond that mediates the interactions with the AB loop, between the AB loop and the helical core structure, right?
I see. So more factors involved.
There's a specific leucine. In fact, it's a little bit of a so hydrophobic interactions north and south of that hydrogen bond that hold it in place, and those were pretty stable throughout the trajectory as well. But the answer is to your question, I guess directly, is yes. It could be that would move in the fullness of time, but what we see is that it doesn't, and that hydrogen bond between that serine 53 and that histidine flicks on and off, but for I guess a third of the trajectory rather than or 3/4 of the trajectory that we saw for the other one.
Yes, all right, thank you.
Okay, Michael, I may have a question.
Oh yeah.
Mm-hmm.
Go ahead.
I noticed that when you prepare, it's about purification step. When you prepare the signal complex, you mentioned the complex when you use the GP130 full active domain. The sample for cryo-EM wasn't size exclusion purified. So may I ask more details about this and why this?
Yeah, that's a good question. So essentially, the best grids that we managed to get had as limited processing as possible. So we got to a stage where we were able to create the hexamer simply by adding each of the components together at 1:1.
Mm-hmm.
Well, so essentially 1:1:1 ratio.
Yeah.
And that gave us a good homogeneous sample of hexamer.
Okay.
And loading that straight onto the grid was essentially the best. That gave us the best grids. So we essentially just removed the size exclusion step from the process. We can size exclusion purify it. But it just turned out that extra processing step, reduced the quality of what we were looking at.
Oh, okay. It's really inspirational. So I think for my preparation of the complex, I may use the same math. Yeah.
Yeah, I mean, it's always worth a try, as long as you have a really good estimate of the concentration of each of your components. They can be put together at a 1:1 without size exclusion. Sometimes it depends a little bit on your system. Sometimes you have to have excess of one of your components, but
okay,
it's worth a try in the grid preparation workflow.
Okay, thank you so much. So any questions? Okay, so so I'm honored to be on behalf of the center. Thank you, Michael, for your catching talk and your time. And thank you, audience, for your attendance. And just remind, our next session will be on May 9 by Dr. Zhou. And looking forward to see you all.